Here we are at PowerPoint 2.4, movement in and out of cells. And like in this graphic here, I've got a picture of some prison cells. When those doors are closed, air can get in and out of those cells, but prisoners cannot. Open the door up and then people can get through. And this is analogous to a cell that you have in your body, because your cell membrane lets some things through. Other things can't get through unless there's something to help them through, a particular protein. There are a number of requirements that need to get into cells, and there are a number of things that cells need to get out. You can read this as well as I can, but take for example glucose. Heterotrophs need glucose. We have to eat to get it into our bodies, and then somehow that glucose has to get into the cells so that we can carry out aerobic respiration. Once we've done that, we need to get rid of the carbon dioxide that's in our cells. So we have to get that out of the cell, into the blood, and then to the lungs where we can breathe it out. So it really depends on what you are, what you need to get in, and what you need to get out. If we compare the things that different types of cells need, autotrophs don't need oxygen most of the time, but sometimes they do. If their aerobic respiration is higher than their photosynthesis, then they're going to need some oxygen. Because, you know, for aerobic respiration, you need O2 plus glucose. And you're going to turn that into CO2 and water. Okay, so that's happening in every cell all the time. When a plant is in the sun, the photosynthesis really outweighs that. So the photosynthesis is the other way around. It's CO2 plus H2O going to O2 plus glucose. So when it's sunny, there is an excess of oxygen and it won't need any oxygen. Okay, a little bit of that oxygen will go over here for the respiration, and the rest of it comes out into the atmosphere and we get to breathe it. But when respiration is higher, they do need oxygen. So at nighttime. On the other hand, we always need oxygen. We're always doing aerobic respiration and we don't get to photosynthesize. Carbon dioxide. Autotrophs need carbon dioxide during the daytime when they're doing photosynthesis. During the daytime, photosynthesis is greater than respiration, so they've got an excess of oxygen, but they're running low on carbon dioxide. We really don't want carbon dioxide at all. In fact, we can't breathe it. If we are stuck in a room full of carbon dioxide, we will die. We also need nitrates and nitrites. They have nitrogen in them, and we need that nitrogen for our amino acids. Phosphates. We need phosphates in our nucleotides. The phosphate group that forms part of the backbone of DNA and RNA. We need it for phospholipids, which are in our cell membranes, and we need it for ATP, adenosine triphosphate. So we learn about nucleotides right at the start of the term. We've learned about phospholipids recently, and we've learned about ATP recently. Calcium, plant cell walls need it. It's also in heterotrophs. We need it as a cofactor for enzymes, to help enzymes. We also need it if we're a vertebrate. We need it for our bones. Okay, so you could also say that we need calcium for bones. Other inorganic nutrients like sulfur and iron needed for synthesis reactions, so making various proteins and so forth. That sulfur there is very important in protein structure. At the level of tertiary structure, because sulfurs form bonds between each other, and so if there's a sulfur in one amino acid and a sulfur in another amino acid, they're often attracted to each other and form a bond. Iron, I'm sure you can think of other reasons we need iron in our body. We need iron in our blood to carry oxygen. Not so much the plants, they don't need iron for that. Organic compounds. Autotrophs don't need any. They make their own. We need some. We need, what do we need? Where are we down here? We need glucose, which we have to eat to get some amino acids, and lipids that can't be made in the cell. The amino acids that we can't make are called essential amino acids. So sometimes you'll see supplements advertised as an essential amino acid, or a food might be advertised as having essential amino acids in it. That's because we can't make them. There's a whole bunch of them we can make, but there are some we can't. Looking at the outputs, there's a similar situation going on here. Autotrophs, when photosynthesis is greater than respiration, so like in the daytime, they are giving out oxygen during the daytime. We don't give out oxygen. Carbon dioxide, they will be giving that out at nighttime, or when it's really dim and they're not doing much photosynthesizing. We are always giving out carbon dioxide because we're always breathing. Autotrophs don't usually give out lactic acid, but we do. We know about fermentation. Your muscles respiring anaerobically will produce lactic acid. Ethanol, not usually happening in heterotrophs. We'd be in a bad way if we did make it. And but from fermentation, anaerobic respiration, autotrophs can. Just to clarify, there's a little problem here. I've just noticed that it says that heterotrophs don't usually produce ethanol, but actually, if you think about it, yeast are actually heterotrophs, and they do produce ethanol. So it's not true for animals. Animals don't, but definitely for fungi, which are heterotrophs. Urea, not normally a thing there. Urea is a waste we get when we break down amino acids. Amino acids have got nitrogen in them. Excess nitrogen build-up in the body is bad. Urea is how humans and other mammals get rid of their nitrogenous waste. Next, we have to explain how the structure of a membrane facilitates different processes of movement through it. So here we go, a bit of revision of the structure of the membrane. We've got all of these phospholipids here. So the phospholipids are really important as a barrier. They don't look like much. They're not very strong. You can poke a hole in them easily enough, but they stop things getting through. And I'll talk about that in a moment. Also, the proteins that are embedded in the membrane, they are very important. You can see up here, there's a word called protein channel, and it's a transport protein. So we know that's going to be involved in transport. So let's have a look at how this all works. If the cell didn't control what was inside it, then we couldn't have all of the reactions occurring in there that we need to happen. So sometimes it has to have some molecules at a higher concentration inside and some molecules at a higher concentration outside. And how easily it gets things through is governed by three different characteristics. So if we look over here, size is the number one thing. If you're small, it's easy for you to pass through that membrane. So for example, oxygen, how many atoms are there in a molecule of oxygen? Just 2 atoms, very small. There's only 3 there. So they get through very easily. And that's how we breathe. These things go through the cell membranes in our lungs to get into our body, and then to leave the blood, they just go through the cell membrane really easily. Lipid solubility. So the phospholipids, are inside, or they constitute the cell membrane. And if something is lipid soluble, it can get through them easier than those things that are less lipid soluble. So it just kind of dissolves its way through. For example, these 4 vitamins here, A, D, E, and K, are lipid soluble, and they can get through that membrane. Because these are lipid soluble, we can also store them in our fat. Things like vitamin C is not lipid soluble, doesn't get into the cell that easily, and also we can't store it in fat. So if you have a lot of vitamin C, your body can't store it. It actually leaves your body through your urine very quickly if you're not using it. Whereas these guys here, you can actually build them up, and you can overdose on them if you have too much. The last thing is electrostatic charge. So you know that the phospholipids have got a polar head and non-polar tails. That means that these guys here are hydrophilic. They like water. If they could like things, they're attracted to water. And these are hydrophobic. So what happens is, because these don't have a charge on them, they're non-polar, they're not charged, anything that's either positive or negative that tries to get through kind of sticks to them, because the charge on these here means they're attracted to anything that's negative or neutral. And if it was a negative ion, it would be attracted to those tails as well. So they might be able to get part way in, but they get kind of caught up in there and they can't get through. So they're small molecules, but they're charged, can't get through. These are small molecules up here, but they don't have a charge, so they can get through. All right, now we're going to look at the 6 main ways that things can get through the membrane. And a bit of revision for you. What is diffusion? Maybe you want to pause the video at this point and just have a bit of a think to yourself. All right, moving on. Diffusion. Here is a definition that you should know. You don't have to know it word for word, but you have to have the gist of it here. Diffusion is the net flow of molecules in a fluid from a region of higher concentration of that substance to one of lower concentration of that substance. So if it's a diffusion of salt, that means there's a high concentration of salt on one side and a lower concentration on the other side. And so we have a movement from one side to the other. The rate of that movement relates to temperature, viscosity of the fluid, and the size of the particles. So if you think back to maybe year 8 science, you know that all particles are in motion all the time. And you know that if you put more energy into a particle, it moves more rapidly. So if you've got a few particles here, and they could be getting across this membrane here because they're moving around a little bit, but they're moving in random directions. So sometimes they will and sometimes they won't. Maybe there's one here that's going through. But if we give them more energy, we actually make those arrows much longer because they have got more energy and they're moving around faster. And so they will move across the barrier faster. Viscosity is a measure of how goopy a liquid is. So, for example, honey has got high viscosity compared to water. Obviously, if everything's a bit more sticky, then it's going to be slower for diffusion to occur. And the size of the particles. If you've got really big particles, they're going to bump into other things. It's going to slow them down. If they're small particles, they're going to get through there much more easily. So the net result of this is if you had a glass and you dropped a bit of food colouring in it, like on the previous slide, eventually it would just all mix together and it would be a uniform colour. If we've got a cell membrane and diffusion's occurring, eventually you're going to reach equilibrium. We've got the same number of, or the same concentration of the substance on one side of the barrier as you do on the other side of the barrier. The other key thing here is that this is a passive transport mechanism. It does require no energy, so no ATP required to do this. A dead cell would do it, okay? Now, I'll stop talking for a minute and I'll show you this video.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Following on from that video, you can see here's a big diagram. Time is on the x-axis across here, just showing you in visual form as well. Here we have some molecules. There are more of them on one side than the other, or a greater concentration. Given time, they start to work their way across and eventually they end up over here. Now, while these things are moving across here, there is still a chance that they can move the other way. There's nothing stopping them, and in fact, they're just as likely to move back across. These ones are just as likely to move back across there as to stay here. But because there are so many more over here, all moving around, they're more likely to go this way. So the net result is that we get a movement over this way until we end up with equilibrium over here. Next, facilitated diffusion. Now the word facilitated simply means help. So this is helping diffusion to happen. And it's a bit like those cell doors at the start of the whole slideshow. If we open those cell doors, people will tend to move out of them if you're in a prison. So large and polar molecules can't get across the membrane via simple diffusion. So they need special, wrong word here, carrier proteins and protein channels to help them. Now they can recognize a specific molecule such as glucose or an amino acid or an ion and help it to move across the membrane. But this won't go against the concentration gradient, it'll only go with the concentration gradient. So here you're looking at this channel and it's opened up and it's letting through these little hexagonal thingies and they're just shooting through because there's a lower concentration on this side. This particular protein here looks like it's going to have to change shape to make it happen, but again that doesn't require any energy. This must embed itself in there, and just the them coming together means that it changes shape and shoots it out the other side. We can see actually here it is at the second step and at the third step, pushing it through this way. Doesn't require any energy. Now this guy's going to say pretty much what I've just said, so I'll play that for you.
In the process known as facilitated diffusion, a special carrier protein with a central channel acts as a selective corridor which helps molecules move across the membrane. These special carrier molecules that form the protein channel bind only to a specific molecule, such as a particular sugar or amino acid. Once the molecule binds to the carrier protein, this protein helps or facilitates the diffusion process by changing shape and moving the molecule down its concentration gradient through the membrane into the cell where it is released. Facilitated diffusion is similar to simple diffusion in that both involve movement of molecules down their concentration gradient, and this movement is carried out without any input of energy. However, in facilitated diffusion, the movement of molecules will only take place if it is facilitated or helped by a special protein carrier in the membrane. Facilitated diffusion can occur in either direction depending on the concentration gradient. If there is a higher concentration of the particular molecule inside the cell, the same carrier protein would then transport the molecules out of the cell.
Okay, so now that facilitated diffusion's out of the way, osmosis. Now this is a bit of a bugbear for some kids because, or students I should say, because people get it mixed up with diffusion all the time. So let's have a look at what osmosis is. So osmosis may occur when there is a semi-permeable membrane. Semi-permeable means that some things can get through easily and some things cannot. Osmosis in cells is the passage through the cell membrane from an of water, just fix that, of water from an area of low solute concentration to one of high solute concentration. I've said in capitals here, so I'm yelling here. Do not mix it up with diffusion. Depending on the concentration of solutes in the solution surrounding a cell, this can mean that water moves in or out. So an example here, I've got a diagram here of a protein complex. These red dots are phospholipid heads for the outside, and these are blue ones are phospholipid heads for the inside. So you can imagine they've got their legs on as well, coming in like this, 2 legs per head. Can't draw very straight with this thing. Aquaporin, aqua meaning water, porin meaning like a pore or a hole, is a membrane protein that acts as a channel in the membrane that speeds the movement of water molecules. So water can shoot through here. It's insulated from the phospholipids. They're not going to hold them up, and it's going to go through this aquaporin. Now, if you have a look at this aquaporin, you can probably see some protein structure here. We've got all of these helices, these alpha helices, which are secondary structure, And the fact that these are in different colors tells me that maybe it's made of different polypeptides as well. So maybe we're seeing some quaternary structure there. All right, I'm going to play you how osmosis works, and then we'll move on.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
Right, now osmosis has a big effect on cells, particularly animal cells. Let's look at what will happen to a red blood cell if you put it into a hypertonic solution. Hyper means high concentration of whatever is around it. So if you put it in there, what's going to happen is that the water is going to, because the whatever it is that's outside that's making the high concentration can't get in, the water moves out to it. And if the water moves out, it leaves these being shriveled up, and we end up with shriveled up red blood cells. Now, for a plant cell, they've got an advantage in that they have got a cell wall, so they actually manage to maintain their shape. However, you would find that they would get a bit more, they'd be a bit less rigid than before, so they'd be flaccid as well. Think of a carrot that you've had sitting in the fridge for a long time. A lot of water leaves the carrot by evaporation, and you're left with like a floppy carrot. If it's in an isotonic, iso means same. Ooh, same, that's really bad writing, same. There'll be some water moving in and some moving out, and the net effect is that you've got no real movement of water, and so it just maintains its normal shape. If you put it into a hypotonic solution, that's low, and to remember hypo and hyper, which is which, hypo rhymes with low, okay, low concentration outside there. So say, for example, if you got these red blood cells and you put them in distilled water. Now what would happen is that water would rush into the red blood cells, and you can see what's happening here. Blah, the whole thing is just popping and bursting. So that would be very bad for your red blood cells. Again, plant cells have an advantage here in that they've got that cell wall around them. It's very unlikely that's going to break. It's very strong. So instead they become turgid, which means really, really stiff or full. And so your carrot, if you have a carrot and you put it into some water, it will tend to take up water and become a little bit crisper. Okay, moving on. Now, those three here were all passive, and now we're into active transport. So it's active because it requires ATP. You need to put energy into this. So protein pumps, for example, use the energy to move some things against their concentration gradient. That's the other thing. If it's moving against the concentration gradient, it must be active. If it's going with the concentration gradient, it's passive. Okay, so we've got, for example, some sodium in here, and we've got excess sodium out here. What happens is this sodium-potassium pump takes in those sodiums, and there's a little bit of ATP comes along and shoots them out. Then 3 potassiums come in here and it changes shape again and comes back. Now, what I don't like about this diagram actually is it's drawn ATP like this, which is just really wrong, isn't it? Because we know that ATP is more like, I don't know, you've got a little ribose there, whoops, and you've got a bunch of phosphates coming off, 1, 2, 3, and what else is on there? We've got, you've got adenine over there, adenine, we've got ribose, and we've got 1, 2, 3 phosphates. So the way they've represented it is not very good here, because it's not like the phosphates are just hanging on the edges, they're in a row. Anyway, I digress. Have a look at this video. You should recognize the animation from the start of previous slideshow. He talks it through very clearly.
The sodium-potassium pump is an active transport mechanism. 3 sodium ions bind to the protein channel, and an ATP provides the energy to change the shape of the channel that in turn drives the ions through the channel. 1 phosphate group from the ATP remains bound with the channel. The sodium ions are released on the other side of the membrane outside of the cell, and the new shape of the channel has a high affinity for potassium ions, and 2 of these ions now bind to the channel. This binding again causes a change in the shape of the protein channel, and this conformational change releases the phosphate group on the cytoplasm side. This release allows the channel to revert to its original shape, and as a result, the potassium ions are released inside the cell. In its original shape, the channel has a high affinity for sodium ions, and when these ions bind again, they initiate another cycle. The important characteristic of this pump is that both sodium and potassium ions are moving from areas of low concentration to areas of high concentration. That is to say, each ion is moving against its concentration gradient. This type of movement can only be achieved by the constant expenditure of ATP energy.
So the sodium-potassium pump is a really good example of a protein carrier. It's a really good example of active transport. Okay, last things that we're going to look at are endocytosis and exocytosis. Endo means in, and exo means out of the cell, into the cell, out of the cell. So endocytosis is the taking in of external substances by the inward pouching of the plasma membrane, forming a vesicle. Okay, so that's how we can get large things in that cannot fit through a protein. And exocytosis is the release of substances from a cell. We call that secretion, when a vesicle from the Golgi body joins with the cell plasma membrane. We've looked at the Golgi body before and what it does. We've seen that in the cell, you've got the cell, you've got the nucleus, you've got the endoplasmic reticulum, which is really hard for me to draw here like this, but it's like that. You've got the Golgi apparatus over here, Golgi. And what happens is we have ribosomes on here, and they make proteins. The proteins get parceled up in here, and off of that will bud a little vesicle, and the vesicle comes across over to here and joins onto the Golgi apparatus, and then it moves its way through here as these mature, gets changed by enzymes, and then it buds off of here and comes to the edge and joins with the end here and releases the proteins that were inside it. Okay, so we've gone through that before, but that's called exocytosis. So have a look at the video and then we'll move on.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use 3 basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
As always with these animations, this is in much more detail than you need to know, but I think it gives you a good idea of all of these different things explained in a different way. Okay, so when we're doing exocytosis, there's actually two reasons the cells do it. One is for constitutive secretion, which is happening all the time. Basically, if you've got a cell this big and you need to grow up and be a bigger cell this big, you have to add to your membrane around the outside, right? So the Golgi apparatus, part of its job there is to get the proteins embedded in the outside there. It might be secreting stuff, but it's also preparing a piece of membrane to add on over here. So this is just joined on here, and any proteins that were embedded in there are now embedded in the cell membrane. Then we've got regulated secretion, which is what I was just drawing a diagram of before, something like that. So this one here is triggered by calcium. Calcium comes along, sends a signal to the cell, and the cell blobs off stuff from here, which joins onto the membrane, and out pops different things. So they can happen at the same time because, the cells are all about multi-purpose. This is actually making the membrane bigger, so it might as well have other proteins in there that it needs as well. There are some subcategories for endocytosis as well. There's lots of words in this slide that you're going to have to learn. I'm sorry about that. There's phagocytosis, which is cell eating. Which we've got down here. So why did I write it? I don't know. If a cell needs to take in a large particle, it can make the membrane form around it. Now, clearly, there's going to have to be some work here by the skeleton inside there. The cytoskeleton is pulling these bits up and around to engulf something. Then that becomes a food vacuole, and what will happen is a lysosome will merge with it to make a larger vacuole, and it'll have digestive enzymes in it that will break apart that thing that's in there. Pinocytosis is cell drinking. So, if it there's some stuff outside the cell that the cell, needs, it can just sort of open up a little pouch, make a little divot, which turns into a pouch, and anything that's out there just ends up trapped in it and comes in. And then later this membrane can dissolve to let those substances into the cell. All right, two things to show you. I'll show you the narrated video first, and then I'm going to show you actual video of a neutrophil, which is a white blood cell, actually engulfing a bacterium.
In phagocytosis, phagocytes are attracted to the area of invasion by chemical products of the microorganism, phospholipids released by injured mammalian cells, or by components of the complement system. The phagocyte moves into the area of invasion and then attaches to the microorganism. The attachment is mediated by a variety of surface receptors, including antibody, lipopolysaccharide, and complement receptors. For example, the C3b component of complement coats bacteria or other particles and then binds to C3b receptors on the phagocyte. This process of coating to enhance phagocytosis is called opsonization. The microorganism is then engulfed by the phagocyte into a vacuole known as a phagosome. Vesicles in the cytoplasm called lysosomes fuse with the phagosome, releasing digestive enzymes such as lysozyme and proteases into the phagosome. The structure resulting from this fusion is called a phagolysosome. Inside the phagolysosome, microorganisms are killed and digested. Finally, the digested contents of the phagolysosome are eliminated from the phagocyte by exocytosis.
So that's showing how a white blood cell called a phagocyte in particular deals with bacteria. Now, things that are taken in by endocytosis aren't always ejected like that. Sometimes it's something that the cell needs. But just keep in mind that that's an example for those guys. So here's a summary. The passive stuff takes no energy, goes with the concentration gradient from high solute to low solute. That is a form of diffusion, whether it's facilitated or simple diffusion. Then osmosis goes from a low solute concentration to a high solute concentration. Again, no energy required. Whereas over here, these things require energy. Protein pumps, any type of cytosis, endocytosis, exocytosis, requires energy to occur. All right, now this bit here tacked on to the end of the PowerPoint is not closely related, but still has got some work to do here. So we're looking at surface area to volume ratio of the cell, and what does that have to do with elephants? Well, have you noticed that elephants are not very shaggy? They don't have a lot of hair on them, right? But lions are. So here's a lion. It's got a big mane, it's got a body, it's got legs, and it's got hair all over it. Okay, keeps it warm at night. Now, why is that? It's because of the surface area to volume ratio of these different animals. Elephants are much bigger. So if you can imagine, if there's a cell in the middle of that elephant, and it's, a cell in the middle of the elephant, and heat needs to escape, heat's got a long way to go. Whereas on the lion, it's got a lot less distance to go. And for every unit of volume that this elephant's got, there's actually less surface area for that heat to escape than the lion has. The elephant's got around this by having huge big flappy ears, which it can use to increase its surface area, and also by not having fur. So how does that relate to cells? Well, let's have a look at this. Here's my elephant, 3 meters by 3 meters by 3 meters. So it's got a surface area of 54 meters squared and a volume of 27. So that's a ratio of 54 to 27. If I simplify that, that becomes 2 to 1. If I've got a lion, 1 meter by 1 meter by 1 meter, that would have a surface area of 6 and a volume of 1, which we don't need to simplify. That's just a ratio of 6 to 1. So the elephant, for every unit of volume, it has got 2 units of surface area to allow heat to escape. Whereas a lion, for every unit of volume, has got 6 units of surface area that lets heat escape. So it needs to be furry. Okay, now this relates to cells because cells need to get in things and get things out. We can see now why there's a difference between volume and surface area as things get bigger is as something gets bigger, the volume increases more rapidly. So the volume's just going to shoot up like that. That one's going up as a square, and that one's going up as a cube. So that's why that is going up quicker. And this is the reason why bacteria don't get the size of your head or a basketball. It's also the reason why we cannot just absorb our food by rolling in it. We need to have a complicated transport system and a digestive system that breaks it down. So the larger a cell gets, the smaller its surface area to volume ratio. Don't get confused though, because the surface area always increases as you get bigger. The volume always increases as you get bigger. Oops. It's just that the volume is going to increase more as you get bigger. So the surface area to volume ratio, how we compare them, that decreases as you get bigger. So now, these last 2 points here from the key ideas, we've actually already covered them in previous slides. Concentration gradients, that's talked about where we're talking about diffusion and osmosis. Okay, diffusion and osmosis. And then the chemical nature of the materials being exchanged, we talked about that before. We talked about small particles, uncharged particles, and lipid-soluble molecules, and how that affects how they move. So large, charged, and/or insoluble molecules require the assistance of active transport. Right, so that's it for this slideshow. What has popped up here, but hasn't popped up here because for some reason it's not loading, is the crash course on cell transport. I suggest you go and check it out on YouTube.